All right, today we're cracking the code of you. We're going to dive deep into the incredible technology of SNP microarrays, which are these powerful tools that let scientists read our individual genetic blueprints with just amazing precision.
So let me just start with a big question. Have you ever really stopped to wonder what it is, deep down in your DNA, that makes you, well, you, different from every other person on the planet? It's a massive question, right? But the answer, believe it or not, comes down to these tiny, single-letter variations in your DNA. And when I say tiny variations, I mean a lot of them. The scale here is just mind-blowing. In the human genome, there are roughly 3 million common spots where our genetic code can be different by just one letter. 3 million. And these tiny differences, they're the secret sauce. They're what determine everything from the color of your eyes to your risk for certain diseases.
Okay, so let's get into what these things actually are. You can kind of think of these millions of variations as tiny little typos in the massive book that is your genetic code. But what's the technical term?
All right, the fancy name is Single Nucleotide Polymorphism, but honestly, everyone just calls them SNPs. SNP. Way easier, right? A SNP is just a change at one single spot in your DNA. Now here's the kicker. For it to be a real SNP, it can't be super rare. It has to show up in at least 1% of the population. That's what makes them so useful. They're common enough to be reliable signposts for scientists to track.
So let's go back to that encyclopedia idea, because it's a really good one. Imagine your entire genome is this huge set of books. A SNP, it's like a single letter being off. Like house being spelled mouse. Now sometimes that typo doesn't really matter. The sentence still pretty much makes sense. But other times, whoa, it can change the entire meaning of everything. And that is why finding them is such a big deal.
So you've got millions of these tiny typos scattered across billions of letters of DNA. How in the world do you find them? I mean, it sounds like looking for a needle in a haystack the size of a planet. You need a special kind of detective, right? A tool built for exactly this job.
And that tool is the SNP Microarray. This thing is an absolute game changer. It's what we call high-throughput technology. What that really means is, instead of looking for one SNP at a time, which would take, forever, this little chip can scan for hundreds of thousands, sometimes even millions, all at the same time. It's a truly monumental leap in efficiency.
Okay, so how does this magic chip actually work? How does this tiny little square of glass pull off such a huge task? Well, it's this really cool mix of biology, chemistry, and engineering, all happening at a totally microscopic level.
It basically boils down to 4 main steps. First, you've got DNA prep. You take a DNA sample, make a bunch of copies, and you stick these little glow-in-the-dark tags on it. Then comes the really cool part, hybridization. You wash this glowing DNA over the chip. The chip itself is covered in these tiny DNA probes, which are like bait for a specific SNP. If your DNA has a match, it sticks. Okay, step 3 is the scan. A laser passes over the chip, and everywhere your DNA stuck to a probe, it lights up. And finally, analysis. A computer sees this pattern of glowing dots and translates it all into digital data, telling you exactly which SNPs you have.
Now, in the world of microarrays, there are really 2 big names you'll hear, Affymetrix and Illumina. Think of them like the Apple and Google of this space. They both get you the same result, but they take slightly different roads to get there. Affymetrix basically prints the DNA probes right onto the chip, kind of like a super high-tech inkjet printer, which lets them pack a ton on there. Illumina, on the other hand, uses these tiny little beads that find their own home in these little wells on the slide. Different methods, but both are incredibly good at what they do.
So, okay, that's the how. It's super cool tech. But what's the why? Why does this actually matter to you and me? Well, this isn't just some toy for scientists in a lab. This technology has a very real, very massive impact on our lives, from our food to our medicine. I mean, the list of applications is huge. We're talking about personalized medicine, where we can use your SNPs to figure out which drug will work best for you. Or think about agriculture, using this to breed crops that are tougher and give us more food. And of course, there are clinical diagnostics to see if you're at risk for certain diseases. It's even used to trace our history as a species, and it's the powerhouse behind these massive studies that link specific SNPs to diseases.
Let's ground this in a real-world example, and it's one you might not expect. We're talking about cabbage. Yeah, cabbage. Back in 2018, a study published in Frontiers in Plant Science used an SNP microarray to help cabbages fight off this really nasty disease called clubroot. And what they found was just awesome. By scanning the cabbage's DNA, the microarray acted like a GPS, pointing them to the exact spots on the chromosomes that were linked to fighting the disease. They found this one spot on chromosome 8 that was a major player in reducing the disease, another one on chromosome 6 that led to healthier roots, and even another on chromosome 8 that helped the plant keep the pathogen levels down. This is the kind of specific, actionable information that plant breeders dream of.
So that brings us to today. What's the state of the union for SNP microarrays? Well, it's a technology with some seriously impressive strengths, but like anything, it's also got its limits. It's really important to see both sides of the coin here.
First off, let's talk about accuracy. These things are reliable. We're talking better than 99.5% accuracy. That's just fantastic. It's why doctors and scientists trust the results so much. But of course, there's always a trade-off. On the plus side, they're super fast, they're relatively cheap, and they're great at what they do, especially for finding things like copy number variations. The big but, the main limitation, is that you can only find what you're already looking for. The chip is designed to find specific, known SNPs. It's not going to discover a brand new mutation that no one has ever seen before. For that kind of work, you have to bring in the heavy hitters, like whole genome sequencing.
So when you boil it all down, SNP microarrays are like taking an incredibly detailed, high-resolution photo of our genome. It's not the whole movie, but it's a vital, powerful snapshot. It's a huge chapter in our ongoing quest to read our own instruction manual. And that leaves us with a pretty mind-bending question for the future, doesn't it? What happens when we can read not just the snapshots, but the entire book? Every single letter of every single person's genetic blueprint. What will we discover then?